Okay, I'm delighted to be here. This has been a very, very stimulating symposium, and I'm grateful for the organizers for going to the effort to put this colloquium together. I took my mandate very seriously. I'm going to talk about some recent developments in comparative molecular biology bearing on the subject of human brain evolution. And I should preface these remarks by saying I'm not a molecular biologist. I'm a physical anthropologist and a neuroanatomist. I do sometimes consort with molecular biologists and have been involved for a number of years in collaborations with people doing comparative genomics. And so I come to this field not really as an insider, but as an interested and sometimes bemused observer. So when I say that I'm talking about human brain specializations or the species-specific features of the human brain, another way to think about that is I'm interested in what happened in the human lineage in the 6 to 8 million years or so since we diverged from our common ancestor with the African apes, in particular chimpanzees. I just put some representatives of chimpanzees and humans there. The chimpanzee there is literally the chimpanzee. That's Clint. He's the Yerkes chimpanzee who was genomed. And the dapper fellow above him is Edward Drinker Cope, who was a very eminent 19th century paleontologist, a famously patrician and arrogant man who volunteered to serve as the type specimen of Homo sapiens. Now, as will become even more clear, I think there is an awful lot we don't know about human brain specializations. There's really only one fact about the human brain that everyone agrees on, and that is our brains are freakishly large. And most of this enlargement has occurred in about the past 2 or 2.5 million years since the genus Homo emerged from the genus Australopithecus. And the differences in brain size between humans and chimpanzees can be seen particularly dramatically in appropriate MR images. The chimpanzee below, his tongue is about as big as his brain. And that, I think, tells you something about what's important in chimpanzee life. Now, one can easily imagine that there are lots of differences in the brains of humans, chimpanzees, and other primates. We know from comparative anatomy that there are all sorts of ways that brains can differ, even among fairly closely related taxa. So, for example, we know the cerebral cortex varies across species in the number of areas that are present, the number of structural divisions. We know that there have been changes in long-distance patterns of connectivity, the way cortical areas are organized into functional networks. We know that there are lots of changes at histological and cellular levels. We just actually, since 1999, we've had the first demonstration of cellular and histological specializations of the human brain. So we know these things must exist. And one thing that I think probably most people have never really thought of very hard is the possibility that there are differences at physiological levels, that the way the cells work, the way they make synapses and break synapses, and the way they make action potentials and do local computations might be quite different. Neurobiologists tend to think that cortex is cortex pretty much wherever you find it. It's got a basic uniformity of organization. It ought to be doing pretty much the same computations in any mammal that you look at. And it's certainly an idea that was very prominent in the 1970s and 80s. I think we have some data now that suggests that humans are rule breakers in this regard. Well, it's useful to ask why we know so little about human brain specializations. And this was a point that was made very forcefully in a broadside published in Nature 20 years ago by Francis Crick and Ted Jones. And they said, all these years we've been studying animals. We really don't know very much about human brains. We don't have very much direct information about the structural organization of the human brain. All of the techniques we neuroscientists use to look at brains in detail, the tracer injections that we use to track connections, the microelectrode techniques that we use to record for neurons, now the genetic manipulations that we do. All of these things are invasive or terminal procedures, and we can't do them in humans. So a lot of what we think we know about humans is actually what we know about animals, and there's an enormous gap in our knowledge. And I would take that one step further and say, well, if we really want to know about human specializations, we have to have techniques not only that we can use in humans, non-invasive techniques that we can use in humans, but also non-invasive or post-mortem techniques that we can use in chimpanzees and other great apes. This is really essential for identifying human specializations. Now, the situation is not quite as bad as it was 20 years ago when Crick and Jones wrote their essay. Since then, particularly in the last 10 years, we've had the development and application to comparative neuroscience of a variety of non-invasive structural and functional imaging techniques. And this is really the subject for another talk, so I'm not going to belabor this. But I'll just make the point that really for the first time now, with the introduction of diffusion tensor imaging, that provides a way of tracking fibers between regions of the brain through the white matter. We really have the first tool that we can use to study not only to study human long-distance cortical connectivity, but to compare patterns of connectivity across humans, chimpanzees, and other primates. And these studies are very laborious, and the progress is slow in part because we're still working out the techniques, particularly the analytic techniques for making sense of the data, and something else that's going on in genomics. But these are methods that offer a great deal of promise for understanding the organization of human cortical networks. Now, the situation is difficult in neuroscience. When we talk about the functions of the human brain at a psychological level, the situation is perhaps even worse. There is very little agreement among people who study who do comparative studies of ape and human psychology about exactly what the differences are between us and them. I think most people today would accept that human language is a human specialization. It's a unique adaptation of our lineage. And certainly people have proposed other kinds of differences in the realm of social intelligence, the ability to represent things symbolically, and also the representation of unobservable states and processes. So for example, the work that Daniel Povinelli has done on the evolution of theory of mind in humans. But it would be wrong to say that there's a consensus in this area. There's tremendous disagreement in this area. And the disagreement is very deep. There's a lot of intellectual and philosophical baggage that people bring to this subject. So on the one hand, there are people who think that the mental gap between humans and other animals is enormous and profound. And you can get a sense of this by asking yourself, do you think a chimpanzee could conceive of an invisible world populated by divine beings that oversee and sometimes interfere in human activities, as, for example, Raphael could when painting his mural? That's a remarkable sort of abstraction. And I think the intuition that most of you probably have is that this is really something that humans can do and probably other species can't. But there are people who don't really share that intuition or look at things rather differently. So there are a lot of people, and particularly in primatology, who think that the differences between humans and apes is really just skin deep. We are, after all, we think, 98 or 99% identical genetically, at least at the level of protein coding genes. And so the differences between us and them are really just skin deep. And people have, this manifests itself in a variety of ways. I mean, and these are not straw men who think this. So for example, Morris Goodman, who is one of the founders, one of the pioneers of molecular anthropology, was so impressed with the similarities between humans and apes that he wanted to sink the genus Pan troglodytes, Pan paniscus, the chimpanzee and bonobo, wanted to simply put them in the genus Homo. And there are a number of people who think that the African ape should be included in the genus Homo. There are other people who would acknowledge that there are differences between humans and chimpanzees, but that they're either not very important or they're not things that we should emphasize, because in their view, the proper role of comparative biology is to emphasize the unity among animal species rather than the differences. My colleague, Franz de Waal at Emory, is someone who takes that view. So there are these very, very different approaches to thinking about human specializations, and you need to bear that in mind, I think. The result of this tension and this lack of consensus is that it's really hard to come up with specific, well-agreed-upon phenotypes that are distinctive of humans, that everyone would agree on. Basically, we have a sort of minimal and very uneasy consensus. Okay, we've got a big brain, and most people would acknowledge that human language is uniquely human. And then we're either more intelligent or we have advanced cognition or whatever those things mean. There's something different about our cognition, but really what it is in any sort of psychological detail, that's entirely up for grabs right now. So, now, enter a new tool into this debate, or actually a set of new tools, the techniques of comparative molecular biology. And from my point of view, what I think is really interesting about these techniques is not so much that they can help us to understand the nature or the origin of phenotypes that we know of, but rather to help us discover phenotypes, human specialized phenotypes that we didn't know exist. This is something I call phenotype discovery. So now it's interesting when new techniques come into play. They land in a field populated by pre-existing ideas, which may not really be appropriate for these new techniques, and sometimes it takes them a while to find their own way around this. So, a lot of our thinking about the genetic differences between humans and chimpanzees goes back to a very famous paper in Science in 1975 by Mary Claire King and Alan Wilson. And at that time, there was enough protein and even DNA sequence information to be able to show that the protein coding genes of humans and chimpanzees were incredibly similar, about 99% similar. And that just floored them. And they said, how do we account for what they saw as the vast phenotypic difference between humans and chimpanzees with such a small amount of genetic diversity? And the answer they proposed is that changes in a few regulatory genes could result from, could account for this. That if you made the right kind of changes, you could build a very different house with the same set of bricks, because the genetic regulation system is sort of the plan for putting these parts together, and you wouldn't need to make a whole lot of changes. They didn't, at that time, there was not a lot known about gene expression, and so some of their mechanistic accounts seem to us a little thin today. But this basic idea is still very much what motivated people to look at the molecular evolution of humans when techniques became available to do that. Another strong influence in this was the writings of Stephen Jay Gould, which have been alluded to a number of times. Basically, Gould took the King and Wilson notion of a few changes and said, well, look, if you make these changes very early in development, you can produce a really profound morphological change just by changing the rate and timing of development of particular morphological features. And so he thought that he had this neoteny theory of human evolution, which he thought that basically humans are extrapolated infant chimpanzees, that you could have a big head and a flat face if you retained chimpanzee growth rates and patterns much later into development than humans do. Today, most physical anthropologists don't take the neoteny theory seriously because it's when you actually start plugging actual fossils or extinct species into this theory, it doesn't work so well. But this was a very influential idea, and in fact, started the whole field of human evo-devo. So enter molecular biology in this. And of course, molecular biology has a whole variety of standard tools that have been used for quite some time to do targeted analysis of particular genes, say the sequence of a particular gene or the post-translational modification of a particular protein. This is the usual, PCR and protein sequencing and Western blotting. And then there are histological techniques like in situ hybridization and immunocytochemistry, immunohistochemistry. But what's really energized the field has been the introduction of some high throughput techniques, particularly first the gene microarrays, which are still being used. There's now another technique coming along called which goes by various names, next generation sequencing or RNA-seq. This is a technique where you just take a whole lot of mRNAs and sequence them all and count up the numbers of different kinds of transcripts. Make that sound very easy. It's actually an incredibly daunting and bioinformatically intensive task. And then we're now starting to get the application of some high throughput proteomic techniques, particularly mass spectrometry-based techniques. These have had a long and painful birth, and the birth is not yet complete, but we're starting to get some fruits from this. I'll talk about that a little later. In addition to these bench techniques, there are also now a variety of resources available for making sense of these molecular differences. So, for example, we have the databases that have resulted from the various species gene and protein sequencing projects. We have a quite sophisticated set of methods for analyzing the functions of genes, so-called gene ontology, and also biochemical pathway databases that people have made a lot of use of. And then there are some statistical techniques for analyzing the covariation between gene expression in tissues, basically correlational techniques to try to figure out networks of gene interaction. There are a variety of techniques. One of the ones I'll talk about is weighted gene co-expression network analysis, and one of the people in this room, Mike Oldham, was one of the people who pioneered this technique. So from my point of view, what's particularly interesting about these techniques is that some of them give us some insights into phenotypic differences. And so I'm going to emphasize, or I'm going to give you at least my take on what the phenotypic consequences are of the various molecular changes that I'm going to discuss. Now, it's quite interesting that the kinds of changes that have been documented span every level of molecular organization. We know that there was a chromosomal fusion event in human evolution. We know that there's been an enormous number of tandem duplicate repeats generated in human evolution. That it's the result that humans have a fairly large number, perhaps hundreds, of human unique genes. Similar processes going on in chimpanzees. There are chimpanzee unique genes as well. There have, of course, been changes in promoter sequence. There have been changes in protein coding sequences. And note that a 1% difference in overall coding still means a lot of differences in the protein coding genes. About 80% of protein coding genes or proteins in between humans and chimpanzees have non-identical sequences. So even at that level, there's a lot of difference. There are differences in RNA splicing. There's tissue and developmental stage specific differences in expression levels. At the protein level, which is the one, I think, from a functional point of view, is most interesting, but also least well known. There are a variety of post-translational modifications that are known. I think one of the best documented, or the most interesting from my point of view, is the way human brain deals with the beta amyloid peptide sequence, of which is identical between humans and chimps, and yet humans form these higher order aggregates of beta amyloid that don't seem to get formed in chimpanzees. So differences at every level of organization. And the fact is that they're just, even though we're in the early stages of mining these differences, there are a lot of them. They're all over the place. Just about any dimension of molecular biology or biochemistry you want to look at, you can find evidence for human specializations. And in fact, if you just line up all the alignable sequences in humans and chimpanzees, which is a sort of simplistic exercise, but interesting anyway, the difference is not, or the similarities are not really 98 or 99%. They're more like 94% or so. So there is a lot of genetic difference between humans and chimpanzees. What do these things mean? Well, I'm going to talk about a couple of lines of research. I can't really do a really in-depth or comprehensive review of this field because it's already pretty big ones. I'm just going to hit a few highlights and emphasize things that I'm more familiar with than with some of the other ideas. When gene microarrays became available, it became apparent to many of us that this gave us the opportunity to test the King and Wilson hypothesis, or at least explore the genome with that hypothesis in mind. So what's the difference between humans and chimpanzees? Well, there ought to be some expression differences, and the differences should be clustered in such a way that perhaps there are just a very few underlying changes that actually generate these differences in gene expression. And microarrays, I'm sure many of you or most of you are familiar with them. What you do, at least in our hands, is to take RNA and hybridize it to human gene chips. These are mostly Affymetrix chips in the first sets of studies. And they interrogated somewhere around 12,000 genes. So not all the genome, but a pretty good chunk of it. These studies had certain problems. The probe sets, the things that the RNAs hybridized to, were human gene sequences. And that can cause you some artifacts because they're not identical between humans and chimps. There's about a 1% difference or so, or 1 or 2% difference. We could actually correct for that by throwing out the probes that had mismatching sequences. Bigger problem comes when you try to hybridize macaques to these arrays because the sequence differences are much greater. And if you start throwing out probe sets, you end up throwing out most of the data. So the outgroup information in this in the early sets of studies were, I think, quite problematic. There were 3 major initial studies, I think. One by Svante Pablo's group with Wolfgang Inard as the first author, Mario Caceres' study, which is done in collaboration with Carolee Barlow and Dan Geschwin and me, and then a study by Monica Udden working in the laboratory of Morris Goodman, who, as I said, was one of the pioneers of molecular anthropology. And these studies, much of the discussion in these papers focuses on a large-scale pattern of gene expression. So Inard et al. Emphasized that there was a higher rate of expression change in the human lineage than in the chimp lineage, especially in the brain. And Caceres et al. Emphasized that if you looked at upregulation and downregulation in human evolution, there were more changes in the upregulation direction than in the down direction. That's the big red stripe on the left side of the heat map. And then Monica Udden et al. Didn't find either of these things. So there was some disparity between these groups in the interpretation of the broad-scale pattern of change. But if you look at gene functions and gene ontologies, there's actually a fair amount of convergence between them. When we looked at the genes that were up and down regulated, we did both the formal gene ontology and a sort of informal analysis. And what we saw were a lot of genes that and remember, this is adult tissue. We saw a lot of genes that were, an excess of genes that were involved in cell growth and maintenance. And we also an excess of chaperone genes, which can be thought of as neuroprotective genes. Informally, what we thought we saw were a number of prominent changes in expression of genes that were involved in neuronal and synaptic activity. I'm going to talk a little bit more about those later. In the Udden et al. Study, they saw an increased expression of genes related to aerobic energy metabolism and neuronal function. So what you're getting a picture of here is a set of genes that seem to be regulating activity levels of cells in the brain. And given the upregulation hypothesis, we thought that what we were seeing was evidence of more neuronal activity, maybe more synaptic plasticity, but in general, a more physiologically dynamic brain. And given our long lifespan and this physiologically highly active brain, it didn't surprise us to see that there would be changes in neuroprotection. In fact, Gaurav Arora, who was a recent PhD at Georgia Tech, reanalyzed our data and found that there was an excess of gene expression changes in humans in genes involved in apoptosis, which is very interesting. They, in his thesis subsequently, he did some experimental studies which show that human cells are unusually resistant to cytotoxins, much more so than our chimpanzee or macaque cells. And he argued on this basis that this is one of the reasons that humans seem to be, that cancers seem to be much more common in humans than they are in non-human primates. They're actually quite uncommon in non-human primates. Anyway, one of the things that we did, and we thought was very important, and which I still think there's been too little of, is we tried to validate some of these results in tissue. So we had changes in gene regulation where these matched by changes in protein expression, and what would we see when we used immunocytochemistry to stain the tissue for these proteins. And we focused on so-called thrombospondin proteins. And the reason that we were interested in thrombospondins is because Ben Barris's lab at Stanford, the work shown on the upper right, showed that thrombospondins were astrocyte-secreted astrocyte-secreted proteins that were essential for generating new synapses. That these were synapse-inducing factors. So we were very interested in looking at these. We confirmed using PCR and Western blotting that two thrombospondins were upregulated in human evolution. And then we did the immunocytochemical staining. And actually, the humans are on the left, and the staining is denser, which is not surprising. What was surprising is that most of the staining is in the neuropil and in pyramidal cell bodies, which suggests that the gene ontology information about thrombospondin expression from the experimental animal literature was steering us a bit wrong about humans. So one of the reasons it's important not to simply rely on gene ontology databases, but to actually look at the tissue. Another gene that we looked at that sort of puzzled us at first was the gene for carbonic anhydrase 2. And in our paper, in situ hybridization, the figure on the upper right, we showed, and our samples were gray matter samples. So we were sort of focused on gray matter. And certainly CA2 is upregulated in human cortex compared to that of chimps and macaques. But the really interesting and striking finding, which we sort of worried about where exactly we should crop this figure, because this is white matter down here. And CA2 is blazing hot in human white matter. And recently we've shown that even more dramatically by doing immunocytochemistry. Well, this is very interesting. What does carbonic anhydrase 2 do? Well, it's an oligodendrocyte gene. At least in primates it is. It's not so clear in other taxa. It's thought to be involved in a transport metabolome that ports lactate into oligodendrocytes, where they use it for energy metabolism, and they also use it as a feedstock for making myelin. And so there are some experimental and observational studies that suggest that CA2 is upregulated when animals are making more myelin. So our interpretation of this was that humans are turning over their myelin faster. Now, we often think of myelin as being sort of the plastic coating that goes over wires. We think of white matter as being a very static entity. In fact, we're learning that it's very dynamic, that it changes in learning and plasticity much in concert with changes in the gray matter. So for example, here's a study of a person learning a simple motor task, and over time what you find is that there are changes in the microstructure of the white matter that can be measured with MRI, in the diffusivity of water in particular regions of the white matter. And these occur in a very short period of time, days to weeks, after people learn the task. In fact, the changes may occur even earlier. I just don't know. We haven't looked at that time scale yet. So what we've got with thrombospondins, with CA2, and with a number of other genes that we've identified as being upregulated or downregulated in humans seem to be involved in neuronal and synaptic activity and plasticity. Monica Udden's study focused on increases or expression changes in genes involved in aerobic metabolism, which makes a lot of sense from the standpoint of our hypothesis. Actually, she worked with Morris Goodman. Morris Goodman's group, and in particularly Larry Grossman's group, have long studied the selection-driven amino acid changes in molecules in the electron transport chain, which is the foundations of aerobic metabolism. And they've documented a long history of adaptive evolution of these molecules in anthropoid, primate, and human evolution. There are some changes, although they usually emphasize anthropoids as a whole. There are also selective changes in the human terminal lineage as well. Now, it's very difficult to make concrete connections between the genetic data and the phenotypes you can actually measure at the organismal level. One of the places that we can do this is when we're talking about cerebral metabolism. So it might seem intuitive to you that as humans we have really active brains and our cerebral metabolism should be very high. But in fact, in physiology, the rule is bigger things tend to burn cooler and tend to run slower. So physiological rates per unit tissue of an elephant are much, much lower than they are of a mouse. And brains are pretty much the same way. So if you look at rats, they have a very high rate of glucose metabolism, which you can measure in awake animals using PET. Rhesus monkeys, there's also data for baboons. They're a little bit lower. But you would think that humans with our very big brains, we should be down here somewhere. But in fact, the published studies suggest that we have, if anything, even higher per unit tissue cerebral metabolic rates than rhesus monkeys. This is something that needs to be reinvestigated. Anyway, to give you an idea about how the paradigm has shifted in comparative molecular studies from identifying genes for known phenotypes to actually finding new phenotypes. I want to consider the gene for FOXP2. This is a story I'm sure many people are familiar with. FOXP2 has widely been touted as a gene involved in language, or a language gene, although most people are very careful to say, well, it may be a language gene, but it's not the language gene. And this was discovered because there was a family in England that showed an autosomal dominant pattern of language impairment. They seem to have a mutation, and these people have very distorted language, very difficult even for native speakers of British English to understand what these people are saying. They have some grammatical problems. Most of the discussions have focused on their orofacial and speech problems. They also have cognitive problems as well. Their average IQs are in the low 80s. They're thought to have other phenotypic issues as well. These have not been publicized or documented very well. But anyway, it's a very interesting story. Of course, people were interested in this gene because of its possible relation to language. Affected members of the KE family do not show the sort of activation of Broca's area that you would expect to see in linguistic tasks. They also show some morphological changes, particularly a reduction in the striatum and the caudate particularly. So with that in mind, Svante Pablo's group, again with Wolfgang Enard in the lead, wanted to see whether there was any evolutionary change in the sequence of this protein in human evolution. And indeed there are. This is a very conservative gene. It's a gene for a transcription factor. Its sequence is highly conserved in mammalian evolution, but there are two sequence changes in human evolution, and that's very unlikely to have occurred by chance. There are the two changes. But there's immediately a problem when you start thinking of FOXP2 as a language gene. It's expressed all over the body. And one of the problems that you have when you can say that this gene was subjected to natural selection, but you don't know what tissue was the target of that selection. It's doing many things, so we don't really know what the function of the tissue is. Simply the fact that you get a knockout mutation in humans and you have some problems with language doesn't mean it necessarily has any very specific relationship to language. So, interesting thing has happened with this is that people have started doing experimental studies to see exactly what the relationship is of FOXP2 to phenotypes and to biochemical pathways and to morphology. So one thing you can do is you can make a FOXP2 transgenic mouse, which Wolf Enard did, made a humanized FOXP2 mouse. And interestingly, only very subtle differences in vocalization, but some rather dramatic changes in dendritic morphology. Dendrites tend to be longer. There are changes in dopamine and exploratory behavior, and interestingly, enhanced plasticity, particularly long-term depression in corticostriatal projections. You can do other kinds of studies. This is a gene that we think is probably important in development. Well, you can look at what genes are the targets of FOXP2 in, say, a developing mouse brain. This is work from a number of workers, including Dan Geschwin and his colleagues. And you can use chromatin immunoprecipitation to identify the genes that FOXP2 is regulating by binding their promoter sites. And one of the things that comes up is axon or neurite extension. If that's your hypothesis, you can examine it experimentally. You can express human FOXP2 in a cell system and show that, in fact, it does induce neurite extension. Another tack that you can take is you can say, well, the genes that are the targets of FOXP2 binding, what happened to them in evolution? Well, quite a number of them have undergone positive selection. This is another paper from Dan's group. And some of these, if you're familiar with the way people look at or estimate selection in genes, usually what they do is they look at the ratio of non-synonymous to synonymous changes, the so-called Ka-Ks ratio. And numbers about 1 or higher are thought to reflect positive selection. Some of these numbers are about the highest numbers that have been reported for any proteins in the human genome. And a number of these things are homeoboxes and other genes that are involved in morphogenesis. So FOXP2 turns out not to simply be involved in language. It may or may not be, but it seems to have a very strong role to play in brain development more generally. Another approach, a very interesting one, is to make cells with humanized or chimpanized versions of FOXP2 and see what the differences are in the genes that are targeted. And they target some genes overlapping and some genes, only the human protein targets, and some of those genes only the chimpanzee protein targets. So I think I'm going to. Then, with those lists of human and chimpanzee targets, then you can go back into tissue and validate the results and see whether those are genes that are targeted in human tissue by the transcription factor. And finally, we have some new techniques for looking at patterns of expression regulated by FOXP2. This is, again, this is one of these weighted gene co-expression network analyses that Mike Oldham has been instrumental in developing. These are genes that are differentially expressed by human FOXP2, or yes, in relation to human FOXP2. And again, you've got a number of genes, transcription factors, and molecules that are involved in regulating neural development. Exactly what aspects of neurodevelopment they're regulating is still not entirely clear. But the trajectory in this work, we started with a phenotype, the KE family, and then we found the mutant gene, and then we found the wild type gene. And that's the usual sort of pattern of gene discovery. But once we get down to here, then the flow starts to go in the other direction. And we find a human-specific gene change. It's probably not just involved in language. It probably has a variety of other roles to play as well. Now, I want to briefly mention some new techniques that are coming along. I mentioned next-generation sequencing, or RNA-seq. This is a system where you take a sample of the RNAs from tissue and you sequence a lot of them, and then you see how many different kinds of transcripts you've got and how many transcripts from each particular gene in the genome. This technique has the advantages that it's, at least in principle, quite species agnostic. You're not running into the problem that you have when you're trying to hybridize macaque RNA onto human microchips. Also, it seems to be more sensitive or more accurate in measuring the amounts of relatively rare transcripts, and also more sensitive in differentiating abundant transcripts as well. So it has said to have a better dynamic range. We're also starting to get some mass spec studies of proteins, and this is recent work from Sante Pavo's lab looking at humans, chimps, and macaques across the lifespan, which is very important because almost all the other expression studies have been of adult animals. This is across the lifespan. And one of the outcomes of this is that they've identified a set of genes that are, in humans compared to chimps and macaques, upregulated throughout the lifespan. And these are genes that, again, tend to be involved in neurite extension and neuronal activity. I'm sorry, synaptic plasticity in particular, both LTP and LTD. So to sum up then, what have we learned from this enterprise? And again, it's an enterprise that's really very new. It's really just finding its legs. We will have new bench techniques. We will have new analytical techniques. But I think that we can draw some tentative conclusions. First of all, there are a lot of genetic changes in human brain evolution. Some of these clearly involve embryonic development. But one of the questions that we'd like to know is, why do we have bigger brains? And we really don't know why that is. We don't know what the mechanism is for generating more neurons. Most of the modifications that are currently documented probably affect adult physiology. And I think that's really surprising. I don't think that people really expected that there would be something different about the neuronal machine and synaptic machinery of humans compared to other animals. Again, evidence for enhanced neuronal growth and neural activity, increased rates of energy metabolism, and also probably enhanced neuroprotection as well. Again, that would make sense for a very long-lived animal with a very high rate of aerobic metabolism. You'd want to be well protected from oxidative stress. What we haven't learned, again, we haven't learned why it is we have big brains. We don't know those genetic mechanisms yet, although there's a project beginning or proposed that would use human and macaque fetal tissue to get at this question. We don't really have a good developmental map of changes in gene or protein expression. We don't have very good regional maps for different parts of the brain either. We need those. We don't really have the genetic correlates of human anatomical specializations, which we'd like to have. And finally, one thing that would be very interesting to have and is very hard to get is an understanding of the chronology of genetic changes that's occurred in the 6 to 8 million years since we diverged from chimpanzees. Of course, we don't have fossils DNA except in a couple of very rare and fairly recent cases, the Denisovan hominids and the Neanderthals. But it's possible, it is possible in principle to date certain kinds of genetic changes based on the pattern of amino acid substitutions within, or the pattern of nucleotide substitutions within the gene. So it's not out of the realm of possibility, although it's not something that we can do right now. And with that, I'll finish.